Good afternoon to all.
Afternoon. Yes. First of all, let me express my deepest gratitude to all of you for being here and to Navodaya School for asking me to give this talk. And I'm essentially going to talk about this particular pandemic that we are facing. But more importantly, I'm going to talk about the virus and what the virus has been doing, how it's spreading, and so on. So that's essentially what I'm going to talk to you about. The first point that I wish to make is that we are talking about a virus, and virus is not a living organism. It cannot live by itself in the sense that even if it is left alone, it cannot reproduce, it cannot make copies of itself. So what does it need for it to survive and for it to live? It needs a host. It needs to get into another living organism's cell. And that's what this virus does. It infects other people, it gets into the cell. And once it gets into the cell, I'll describe what it does. It gets into the cell and then it propagates itself. It infects that person and that person in turn infects somebody else. And that's how the virus propagates. So the first thing that the virus must do is to find a host. In this particular case, the host is a human. That's what we are concerned about. It reproduces, makes copies of itself, and then when that person infects another person by sneezing or however, then that particular person gets the virus and the virus again gets into the host cells itself. That's how the propagation takes place. So this is what a virus is all about. The structure of the virus is very simple. It has what's called the DNA or an RNA. It's a string of alphabets. In the DNA, you will have alphabets A, T, G, and C all strung together in a linear fashion. So it's really like a sentence, but it's not naked. It's covered with what's called a lipid, which is really a fat. So it's covered with fat. This particular nucleotide sequence is covered with a fat. So in DNA, like I said, it's A, T, G, and C. In RNA, it's A, U, G, and C. But it's essentially the same structure, a linear structure that's covered with. Point is that these are linear structures, and these linear structures are actually covered with a fat. So this fat can easily melt, so to say. And one of the ways that to melt the fat is to wash your hands. So essentially, the soap water does just this, that it melts the fat and it exposes the RNA. So this is this particular virus that we are talking about, which is the SARS-CoV-2. SARS-CoV-2 is essentially an RNA virus, meaning that it has a linear string of A, U, G, and C covered with a fat cover. And this particular virus is a very long virus. It's a large virus, meaning that the number of alphabets that it has in its RNA is 30,000 alphabets. That's very long for a virus. There is also another advantage that this particular virus has, and I'll be slightly technical. This particular virus is a positive stranded RNA virus. By that, what it means is that normally a virus, an RNA virus, if it is not positively stranded, it needs to go through one more step for it to become for it to make certain kinds of proteins. This particular virus does not need to go through one more step. It's all ready to make a protein. And these proteins are building blocks of life. These proteins help in lots of various kinds of things. And this virus also can produce these proteins once it enters the host cell. This is what it is. It's a positive stranded RNA virus. It's a long RNA virus of 30,000 alphabets. One of the questions that we have, and you have read this in newspapers, etc., where did this virus come from? It came in December. We got to know of this virus in December of 2019. So it's just been about 6-7 months ago that this virus suddenly made headlines. And we were all curious, where did it come from? So people started searching, and the way that we make these searches is to find similar viruses in other animals. So we speculate that this virus came from other animals, and we need to find out the source. What other animal is there? So one of the sources that we know, one of the popular sources is a bat. Now, why a bat, we still don't know, but bat is home to many different kinds of viruses. At least we know of 60, 70 different kinds of viruses that can reside in a bat. And then sometimes the bat will transfer this particular virus to another species. And that's how this is called a zoonotic transfer. So that's how it crosses the species barrier and starts infecting another species. So the first suspect was a bat, and bats do have coronaviruses. But how do we know that this particular coronavirus came from the bat? One of the ways of doing this is to sequence the RNA, which essentially means that you read the sentence of a coronavirus in the bat and you read the same sentence of the coronavirus in the human and see how identical these two sentences are. So essentially, you sequence the RNA of the bat coronavirus and we sequence the RNA of the human coronavirus, and we tried matching them letter by letter and found that these are 96% identical. So the bat coronavirus was 96% identical to the human coronavirus letter by letter. So of these 30,000 letters, 96% of those letters were exactly matched between the bat and the human. There were other coronaviruses that had also been sequenced, and the match was not so good. So we reasoned that most likely this particular coronavirus came from the bat, and the bats were the ancestors of the human. The bat coronavirus is the ancestor of the human coronavirus. Remember that they are not exactly identical. There are 4% of the alphabets are different. So there are certain properties in the human coronavirus that's slightly different from the bat coronavirus. But the bat coronavirus is the ancestor of the human coronavirus. Now, it's also known that sometimes this particular transfer to humans don't take place directly. There is a medium, there is a mediator, and that mediator is another animal. So this was known with respect to other viruses. So we started searching. Many people started searching, can there be another mediator through which it came from the bat to that mediator and from that mediator to the human? That's possible. So people started looking for it, and it was an accidental discovery. I'm sure that all of you know that this particular coronavirus was first identified in China in a place called Wuhan. And the way that it was identified is that Wuhan has a very large market dealing with seafood market it's called. That market also deals with various kinds of other animals. And these people, they butchered the animals, they cut the fish, and so on and so forth. So one of those people who was handling animals actually fell sick with very similar symptoms that we see for COVID-19 today. And that particular worker in the seafood market did not actually die, recovered, and the person is living today as well. So, but from that person, the lungs were aspirated, and so from the lungs, fluid was collected, and from the fluid, the virus was isolated, and the virus was sequenced. Now, the kind of symptoms that we have, the humans have, what was found not very far from Wuhan, about 150 miles away from Wuhan, they found that there were two pangolins. Pangolins are anteaters, scaly anteaters. Two pangolins, these are another kind of animal. These two animals were found dead, and they had all kinds of froth coming out of their mouth, and they died. So when froth comes out of the mouth, one of the suspicion is that lungs were infected with something. And so what they did was to look at the fluid that was accumulated in the lungs, and they isolated, and lo and behold, it was a coronavirus. Now, they also sequenced this particular coronavirus and matched letter by letter with the human coronavirus, with the bat coronavirus, etc. So we think that the bat transmitted the coronavirus to this anteater called pangolin, and the pangolin in turn gave this or transmitted this coronavirus to human. So it is important to know the cycle by which these coronaviruses get transmitted, because not for this, but for a later period of time, for when another virus starts to get transmitted, you can probably break the cycle, break the chain of transmission. So it's important for us to know, how these viruses get transmitted from one species to another. So that's what we do. So I've told you a very important thing, which is that we compare RNA sequences, and the two RNA sequences, the two coronaviruses that match the highest, we believe that that's the ancestor of the coronavirus that we're talking about. So the human coronavirus matched the bat coronavirus significantly well. And so we believe that that's the ancestor of the human coronavirus. Now, we will not talk about other animal species anymore. We will now concentrate on the humans. So we talk about the human coronavirus, and there was this particular one coronavirus that was sequenced, found in Wuhan seafood market and was sequenced. And that particular coronavirus was called the O coronavirus. So it was called type O. Don't worry about how these names come about. So these viruses are given names. So this coronavirus, the first coronavirus was given the name O or label O. So that particular O label had a specific RNA sequence associated with it. This virus, the human coronavirus, the one that we are now worried about, that's spreading so fast and causing some number of deaths. It's caused a large number of deaths because it has infected a very large number of people. But if you want to look at it, if you want to look at the virus, the one thing that can be said of this virus is that it infects with great efficiency, but it does not kill. It's a very kind virus in some ways. Now, this might actually come as a surprise to you that I'm calling this virus that we are all so worried about as a kind virus. Why am I calling this virus to be kind? This virus, of the 100 people that it infects, this virus kills about 2 or 3 people. If you look at other human coronaviruses, and there are many human coronaviruses, most of them are actually not pathogenic. They don't cause any harm to humans. But there are at least 2 other coronaviruses that we know that can kill and that can infect. And there were outbreaks of those coronaviruses earlier. The first coronavirus outbreak that was pathogenic, meaning that it killed people, was in 2003, and that outbreak took place in China. That coronavirus was called Sarsko V. We were not sure that there would be another similar coronavirus. Now it's called Sarsko V2. So that original one may be called as Sarsko V1. So that Sarsko V1, of the 100 people that it infected, it killed about 11 people on an average. Compare it with SARS-CoV-2, it's killing about 2 or 3 people. SARS-CoV-1 killed 11% of the people that it infected. Then there was another outbreak. That particular outbreak, the SARS-CoV-1 outbreak, was contained in China, and it didn't spread very much. And I'll talk about how you can prevent spread and how you can measure whether it's spreading fast or not. I'll come to that in a minute. There was another subsequent outbreak, and that outbreak took place about 10 years later in 2013. And that outbreak did not take place in China, and that outbreak took place in the Middle East. And that coronavirus outbreak is called the Middle East Respiratory Syndrome coronavirus. It's called the MERS coronavirus, Middle East Respiratory Syndrome coronavirus. That was a dangerous virus. Of the 100 people that particular coronavirus infected, it killed on an average 34 people. So remember the 3 numbers: 34 of 100, 11 of 100, and 2 or 3 of 100. So I'm sure that you wouldn't blame me for calling this particular coronavirus as a very kind coronavirus. It does not kill many people. So that's the reason why I think that we should call this a very kind coronavirus. We are very worried because it's spreading very fast. It's infecting a large number of people. So one of the things that, it's impacting on health of communities, right? It's impacting on public health. And public health experts must know, is the virus still spreading? Is the virus not spreading? Is it contained? Is the pandemic going to recede? It's going to come down soon. How do you find these out? There are multiple ways that we find this out. It's not a matter of one's opinion. We need to get a quantitative way of measuring so that given a piece of information, given some piece of information, you and I will come to the same conclusion. That is the way that we should deal with science. Given some data, given some information, we need to draw conclusions from that information such that it is not dependent on the person who is looking at the data. So we need some objective ways of dealing with it, and one objective measure, you may or may not have read this, one objective measure of finding out whether whether a virus is spreading or not is called R naught. R naught is, let me interpret R naught first and then tell you how it is calculated. The interpretation of R naught is that R naught has a value like 2 or 3 or 5, whatever. The interpretation of that value is that it is what is the number of people that's being infected by one infected person. So you have a number of uninfected people and you have one infected people. What is the number of people that this one infected person is infecting, right? That's called R naught. So if you can imagine that if I have R naught equal to 5 versus R naught equal to 3, R naught equal to 5 means that an infected person is infecting on an average 5 people. R naught equal to 3 means that an infected person is infecting on an average 3 people. So when it's infecting 5 people on an average, it's going to spread very fast, at least faster than when R naught is equal to 3. This is clear, right? So one way to find out whether the virus is still spreading, is infecting many people, is to estimate R naught. I'll come to how you estimate R naught. When R naught is equal to 1, that means what? One infected person is infecting one uninfected person. That means the virus is not spreading. It's reached a plateau. It's reached a stage when it is not infecting more than the number of infected people that there are in the community. When R naught becomes less than 1, let's say that R naught becomes 0.6, that means that if there is one infected person, it's infecting less than one uninfected person, which means that if there are 100 infected people, those 100 infected people are infecting 60 uninfected people. So 100 versus 60, that means the overall, the spread of the infection, the number of infected people in the community, in the population is going to come down. So R naught is a good measure by which we find out whether a specific virus or any pathogen for that matter is spreading in the community and is increasing in its spread or not. Right now, I don't have an exact number for R naught. Right now in India, the value of R naught is estimated to be about 3. It varies from one location to another. It's not the same in Mumbai. It's not the same in Kolkata. It's not the same in Bangalore. There is a variability, but on an average, I'm told that it is between 2 and 3 now, which means that the virus is still expanding. Its spread is expanding. And when it becomes less than 1, then it will contract, meaning that the spread of this pandemic is going to come down, and we can then move around freely. We can go to a birthday party, we can go to a marriage party, etc. But right now, that's all no, because one infected person is infecting more than one uninfected person. So we have to be careful. This is how we measure the spread of this is a very popular measure, very effective measure. I'm not going to get into the details of how this is done, but essentially it has 3 components. One is what is the total number of uninfected people in the community. If an uninfected people comes in contact with an infected person, what is the chance that the virus will spread to the uninfected person, and how many days does it take for an infected person to show symptoms? So these are the kinds of 3 components that it goes into the estimation of this particular objective measure called R naught. The only thing that we have handle on, the only quantity that we have handle on in these 3 terms that there are in R naught is the total number of uninfected people in the population. These uninfected people are like susceptible people. So that means that an individual, if exposed to the virus, will become infected. So that's called a susceptible person. We can reduce the number of susceptibles in the community, in the population. How do we do that? One is a natural process, which is that I get exposed to the virus, so I gain some immunity from the virus, and once I recover, I'm no longer susceptible. I'm removed from the pool of susceptible individuals in the community. The other way to do this is kind of artificial. I don't get exposed to the virus, but I have a mixture, or I have something that will mimic, that will be like a virus, that will mimic a virus, and that's called a vaccine. So if you have a vaccine is not a virus, it doesn't infect you, but the body's immune system recognizes a vaccine such that it mounts the very same kind of protective measures that the body will take. The immunological system in the body will become awake to that particular kind of virus. But unfortunately, so a vaccine improves the immunity against a particular kind of virus, and then you're no longer susceptible to infection by that virus. So if there is a vaccine that's available, we can make a large number of people non-susceptible, large number of people who are not yet infected. Vaccinating them, we can remove them from the pool of susceptible individuals in the community. So vaccines are very helpful, because if you reduce the number of susceptibles in the community, then R naught will become less than 1. So that's one way of making R naught less than 1. And if R naught gets less than 1, then the virus is not spreading in the community, and you have contained the pandemic. You have reduced the impact of the pandemic. Unfortunately, for SARS-CoV-2, we do not have a vaccine yet. There is a huge amount of work going on, really huge amount of work going on. Lots of money is being pumped into identifying or discovering a vaccine against this particular virus. But researchers have not been successful. Many drug manufacturing and pharmaceutical companies are on the job, and every day we hope that there will be a vaccine, but it hasn't come through yet. It's been over 6 months that this particular virus was first identified, and people got into the task of finding a vaccine. Hasn't been found yet. That doesn't mean that scientists are idiots. That doesn't mean that scientists are sleeping. They are working day and night. It's not easy to find a vaccine against a specific virus. A vaccine must not have any side effects, but oftentimes, you can kill the virus, but you also kill, or at least cause damage to the person who you're injecting the vaccine to. So you need to make sure that there are no side effects of this vaccine, and it's actually protective without any side effects. So it's not easy to find something like that. People are working hard. They have not yet been successful, but we keep hoping every day that there will be a vaccine, in which case, if I'm not infected, I'll take the vaccine. I will protect myself against this particular virus. And R naught will come down, because the pool of susceptibles is then going to shrink. And once the pool of susceptibles shrinks, the virus can find no other person to infect, and therefore the virus will die a natural death. Remember, I told you right in the beginning, if the virus cannot find a host into whose cell it will go and start reproducing, then the virus is dead. So the point is that if you make everybody immune to the virus, then the virus will no longer find anybody else to infect, and therefore cannot find a host cell, and therefore the virus will die a natural death. The pandemic will be gone from the face of this earth. But again, it's a long haul. It's still not come. Many people are getting infected. Many people are getting infected without showing symptoms. So I right now don't have any symptoms of COVID-19. But I may actually be infected with SARS-CoV-2, which means that it is good for me, because I'm not showing any symptoms, and I'm becoming immune to the virus, and therefore I'm no longer susceptible. However, I'm not showing any symptoms. These people are called asymptomatic. I'm not showing any symptoms of infection, and there's another person who is also not showing any symptoms of infection. I come in close contact with that person, and I infect that person because I'm infected but do not have any symptoms. So asymptomatic individuals who are asymptomatic and infected themselves are good, because they don't have to get themselves hospitalized, but they are infecting a large number of people, and that's dangerous for the community, because we can't be sure that if an asymptomatic person is infecting another uninfected person, then that uninfected person will also remain asymptomatic. This is not guaranteed. If I am asymptomatic, I transmit the virus to another person uninfected. That person may come down with, may have to be hospitalized, may be dead. So we need to be very careful about this asymptomatic infected people as well. But we can't detect them, so can't easily detect them. So it's kind of a difficult situation. This whole business against a phrase that you may have seen, you may have read, it's called herd immunity. What that means is that if a large number of people in the community are infected or have become immune to the virus, and like I said just now, that you can gain immunity by one of two ways: through a vaccination or through a natural infection. We don't have a vaccination right now against SARS-CoV-2, so we have to get naturally infected for us to gain immunity. So supposing I have 1 million people in the community, a question is, how many of these 1 million people must be naturally infected and therefore must have gained immunity in order that the virus will come down, in order that the pandemic will come down in its spread, in its prevalence. We can do some mathematics, and one can show that if you have about 50% of the population immune to the virus, then the virus will be more or less contained, will be more or less stable. When the fraction of people in the community who are immune to the virus or who have gained immunity becomes like 67%, then you can be sure that the pandemic will come down. But for 67% of the Indian population to gain immunity will take a long, long, long time. And of course, of those 67% of the large population that we have, even if 2 or 3% die, you'll have a very large number of deaths. Can't be avoided. We don't know any way of avoiding by wearing a mask, by using sanitizers, by keeping ourselves physically distanced. Essentially, what we are trying to do is to elongate the process such that if lots of people come down with the infection and disease, then there will not be any hospital beds to provide treatment. There will be no doctors to provide treatment to us. So by stretching this process of infection, the one good thing that we are doing is the health system is not overburdened. If the health system is overburdened, then the health system cannot take care of people who are infected and have symptoms. So in some ways, stretching it is good, but people are already fatigued of wearing a mask. If you go out on the road right now, everybody has a mask, but it's not covering the nose and the mouth. It's on the chin. So that's like not wearing a mask at all. So the point is that people have gotten fatigued. It's not fun to wear a mask. So you can't talk very well unless you have a particular kind of mask, and so on and so forth. So people are actually getting mask fatigue, and so the virus is likely to spread faster, which is really dangerous. So I've talked about characteristics of the virus. I've talked about how the virus spreads and how do you know whether the virus is spreading or the spread is coming down. The last part of my talk is about how this virus has evolved and what is it that we see of the evolution of the virus. What do I mean by saying that the virus has evolved? Remember that I said that when the first person was found infected with this coronavirus, that person's virus was isolated, and the virus was sequenced, and we called this sequence as O. That when the virus spreads, when time elapses, when time passes by, there are changes that take place in the RNA of this virus. These are natural changes. Nobody is deliberately making these changes. These changes happen just naturally. And so if you take today 2 coronaviruses, they will not have exactly the same sequence. They will not have the identical sequence. There will be few that will be few alphabets or few nucleotides that will be different between these two sequences. So this is so if we collect a very large number of sequences of a very large number of coronaviruses and we compare them, we can find that there are some coronaviruses or some of these sequences are very similar. Some of these sequences are not so similar. So we now try to do some statistical analysis of these kinds of sequences. And what we find is that you can group these sequences or group the viruses into right now into the original was one group, the O group, and then subsequently there are 10 more branches of this tree that has evolved. So it's like a tree, it's like a growing tree. As the virus is, as time is elapsing, as the virus is spreading, the virus is also acquiring new changes in its RNA called mutations. So new mutations are accumulating. So it's like a tree with its root as O and with multiple branches. There are right now 10 different branches of this particular tree. The branches are also given names, and they're given very peculiar names. So for example, one branch is called A2, the other branch is called A2A, then there's a branch called B, there's a branch called B1, and different subtypes, and the original subtype which is called O. And so there are totally today, if we look at the pool of coronaviruses in the world, we will find that there are 11 different subtypes of this coronavirus. Why are we interested in these subtypes? Well, natural curiosity. How many types of these subtypes of this virus are there? So there are 11 of these subtypes, and they are slightly dissimilar, but they all belong to the Sarsko-V2. They're all coronaviruses that that infect us in similar ways and so on. We noticed, we meaning a few of us noticed something very, very interesting. So this coronavirus arose in China, and it moved from China to Hong Kong to Europe to America, right? It moved. Now it's all over. It moved to India as well. Initially, the move was through some travelers who traveled from China to other parts of the world. They carried the virus, and then the virus spread, and in that particular, wherever they went, then they infected other people, the virus spread, etc. What we found was that, for example, if you look at Europe, in January, in the beginning of January, you found that from the sequences that were available, it was found that there was a diversity of different kinds of different subtypes. End of January, the diversity had increased, but very soon thereafter, one particular subtype, and this subtype is called A2A. This particular subtype was overtaking the other subtypes, as though this particular subtype was a marathon runner and a much better marathon runner than the other subtypes. So this A2A spread very, very rapidly to Europe. It outcompeted, it overtook all of the other different, the 10 other types of viruses, and overtook and spread very rapidly in Europe and also in Americas. So then we got curious. There are these 11 different subtypes of this virus, and in Europe and America, only one particular subtype, the A2A subtype, was able to outcompete everybody else and all other subtypes and spread its wings far and wide. Why so? We looked at other places also, and what we found is that in East Asia, South Asia, India, Thailand, China, Japan, these regions, where people are of a different ethnic group. The Europeans and the Americans are called Caucasoids. They are of a specific ethnic type, and other places, like China, India, Japan, Hong Kong, Malaysia, they are of a different ethnic group. They are not called Caucasoids. So among the Caucasoids, it was spreading very, very fast. In the non-Caucasoids, we found that A2A was spreading, but it was not spreading as rapidly or as much as in Europe or in North America. We got curious. So the first observation was that even though there were 11 subtypes, only one subtype had become dominant in Europe and North America. However, in East Asia and in South Asia, there was still a diversity. So we asked why so. And one of the things that we started exploring, and the virus was known, we knew certain properties, certain characteristics of the virus. So I will tell you two of the characteristics, and I will spend a little time on one characteristic and less time on the other. So the first characteristic is that the virus, like I said, that the virus cannot live by itself, it needs a host cell. So how does it enter the body? It enters the body when an infected person sneezes. It gives out, that person gives out lots of droplets, and these viruses are contained in the droplets. We inhale that droplet and the virus gets into our lungs. This virus predominantly goes and infects our lungs. So it's called a respiratory coronavirus. It infects our lungs. So it gets into the lungs. After it gets into the lungs, what does it do? Does it automatically start reproducing? No, it can't. It still needs to enter a cell. So the first thing that the virus needs to do is that it needs to anchor itself to the surface of a cell, of a lung cell. These lung cells are called alveoli or pneumocytes. So there are technical names, but don't worry about that. It goes and anchors itself, attaches itself to a lung cell. The first thing that it needs to do is to attach. The second thing that it needs to do is, having attached, it needs to break open the cell membrane. The human cell has a membrane. It needs to break open the cell membrane and insert itself and get into the host cell. Only then can it start using our cell machinery. So it first needs to anchor itself, then it needs to crack open the cell membrane, the human cell membrane, and it needs to enter. After it breaks open the cell membrane, it needs to enter this host cell. Then once it enters the host cell, then the host cell has many kinds of machinery inside the cell that will help the virus to reproduce itself. So the first thing is anchor. What helps anchoring? The spike protein, you know, a coronavirus, you have seen pictures. It's like a round ball with lots of these tentacle-like things. These are flares, like the corona flares of the sun. So those are called spikes, and those are actually proteins. And that spike protein goes and first anchors itself to the host cell. This is the protein, and it anchors itself against another protein. What is that protein that is available on the human cell surface to which the virus spike protein is anchoring? That is called angiotensin converting enzyme 2. Don't you worry about the name. I'm just, for completeness, I'm telling you this name. It's called ACE2. And this is a protein that's available in plenty on the surface of the lung cells. And this is where the spike protein will go and attach itself. So two proteins have attached itself. The virus has anchored itself to the host cell. Now it needs to break open the host cell and enter. I'll come to that part of the story in a minute. Why is it that this particular subtype, the A2A, is able to spread itself so quickly and so easily, so rapidly? We looked at this, and very interestingly, this particular subtype, the A2A subtype, has a mutation in the spike protein or near the spike protein of the virus that enhances its ability, that makes it more efficient to go and anchor itself to the host cell. So compared to the other subtypes of the coronavirus, which are also able to attach itself to the host cell, this particular subtype is far more efficient. And it's more efficient because this subtype has a specific mutation that enables it to gain this efficiency. Once it anchors well, it anchors more easily to the host cell. Chances are it will also be able to enter the host cell easily. So this is the reason why we think that the A2A virus subtype is spreading so rapidly and so easily. There is a second part to it, which I have not and I will not describe in any great detail, which is that after it anchors itself, it needs to break open the cell membrane and enter. And even to break open the cell membrane and enter, there are other proteins that are at play. There are other proteins that are required, and these are called proteases. A protease is like a pair of scissors that will cut another protein, so it will cut the cell membrane. These proteases in particular are called serine proteases. It will get a little bit technical. I don't want to get into the technicalities right now, but this A2A is also able to use these serine proteases or get these serine proteases expressed more in some ways such that it's able to more efficiently cut the cell membrane and also enter. So what we believe is that this particular subtype, because of a mutation, is able to anchor itself more efficiently than the other subtypes, anchor itself more efficiently to the host cell, and it's also able to enter the host cell more easily compared to the other coronaviruses, other subtypes of this coronavirus. So that's the reason why we believe that this particular subtype has spread so easily. And not just this subtype, this is also the reason why this coronavirus has been able to has been so successful in its ability to spread so rapidly. So this is what it is. There is, like I said, that viruses are usually dangerous. They kill a large number of people, but this virus is not so virulent. Does not kill as many people. It spreads very well, but does not kill. So we have understood how it spreads, how one particular subtype spreads so well, but there is not very much of research that's going on in trying to understand how it kills, because it does not kill very efficiently. So there is no reason for us to, pay too much of attention to what enhances or what gives it the ability to kill people. It doesn't kill very many people. The last point that I want to make before I end and entertain your questions is that one makes a vaccine against a virus. If there are multiple subtypes, the vaccine manufacturer or the person who is discovering or inventing the vaccine must keep in mind that if it misses out one particular subtype, if the vaccine cannot kill one particular subtype but will kill all the other subtypes, then the vaccine is useless. It must be effective against all subtypes of that particular virus. Otherwise, it will not be efficacious. It will not help protect individuals from the coronavirus that we are dealing with. So there are many things that a vaccinologist, a person who is trying to discover these vaccines, must keep in mind. One is that it must be effective against all subtypes. Second, it must have no adverse reactions against the human who is taking the vaccine. So finding a vaccine is taking a lot of time, but these kinds of findings about multiple subtypes, how it transfers from one species to another, all of this are very useful basic information, and this basic information is used by people who actually are trying to do something practical, like finding a vaccine or finding a treatment for COVID-19. So I will end my talk here. Let me quickly summarize. I talked to you about characteristics of the virus. I talked to you about how we monitored the spread of the virus. And the third thing that I talked to you about was the evolution of the virus, that it evolved in bats, it came to humans. Even within humans, there are now 11 different subtypes, and one particular subtype is able to spread itself very well. And I've given you an explanation as to why this subtype is able to spread itself very well, because there are two facets involved. One is anchoring, the other is entry, and the virus is able to do very well on both counts, and therefore it's spreading very well. We hope that there will be a vaccine very soon, such that in addition to the natural infection, we can get ourselves vaccinated and gain immunity such that we are removed from the pool of susceptibles. And once we are removed from the pool of susceptibles, the virus is going to come down, because R0 is going to come below 1. So thank you very much for your attention, and I'm happy to take any questions.
Good afternoon, sir.
Yes, good afternoon.
As you said, it is most rapidly it is spreading, or those population is more susceptible, genetic composition. I mean, suppose India is having it, so it is around both.
Yes, very good question. I actually hinted to this, right? I said that those people are Caucasoids, and that's where this particular virus was spreading. But I also came to the last part of my talk, and I said that I'm going to cut short. So you've now pushed me to telling you this. Again, I'm not going to, it will become a little too technical. So I will tell you this, that in terms of anchoring itself, we don't find any reasons to believe that there are ethnic differences. But when it comes to breaking open the host cell membrane and entry, this particular virus finds it easier to break open a caucasoid cell than, for example, a mongoloid cell. Why does this happen? It happens because your genome and my genome are not the same. It's because an American person's genome is not the same as a Chinese genome. There are significant differences, and those host genomic differences is what makes this difference in its ability, in the coronavirus's ability to enter the host cell. I'll leave it at that because if I want to describe more, it'll become very technical. But you're completely right. The reason is not in the virus. The reason is in the host. Genomic differences in the host also play a major role in the ability of the virus to enter the host cell.
Thank you, sir. Thank you.
There's a question from a student. Asymptomatic patients, are they totally safe?
Yes, so safe meaning that they will not have respiratory problems, they will not have other kinds of problems that will require hospitalization. So in some ways they are safe. They themselves are safe, but they are very unsafe for other people who are uninfected, because the other people might think that you are uninfected when in fact you are infected, and then you will go and infect them. So it's some ways they themselves are safe, but they are very unsafe for other people.
Good afternoon, sir. How it affects different people with different blood groups?
That has been published saying that the B blood group people are more susceptible to the coronavirus. Again, these studies are very quickly. These studies are based on small numbers of people, so we need to enlarge the sample size and see if that that result will hold up when we increase the total number of individuals. So we need to wait and see. But right now, the word is that B blood group people are protected against the coronavirus. They're less susceptible, not protected, but less susceptible.
Thank you, sir.
Good afternoon, sir. A lot of companies advertising their product and claim that they provide immunity to us, sir. Is it true, sir? And to what extent, sir?
So yeah, immunity boosters are there. I mean, there are certain kinds of food products that can increase immunity. There are also certain kinds of medications that can increase immunity. But again, immunity is a very broad term. Not every kind of immunity will be efficient against this virus. So I really don't know whether, overall gaining of immunity is good. Then you are less susceptible to infection. But whether it protects you against this particular viral infection, I do not know. But it's not a bad idea to increase your overall immunity. Yes, the answer to your question is there are foods and some medicines that increase immunity. So it's not a bad idea to boost overall immunity.
Thank you, sir.
Sir, this you told that asymptomatic person is safe.
Yes.
How long the virus will stay in him?
So usually, even if it's asymptomatic or symptomatic, our current estimate is about 3 weeks. So plus minus a few days, but it's usually 3 weeks that this person will continue to test positive.
How long do you expect a broad speculation about how many months more until you can safely step out?
Many months, primarily because lockdown is coming and going. If you lift lockdown, maybe we can make a prediction. But with lockdown coming and going, you can make no prediction. But all that I can say is that it's going to last a long time. Let's all be prepared to deal with the virus for a long time. And there were talks about rains washing away the virus. Summer coming, the virus will be killed by heat. All of this is there's no scientific basis of these statements. So let's just prepare ourselves, brace ourselves to dealing with this virus for a very long time. And by very long, I mean maybe another year or so.
Sir, kindly accept our thanks, and we are looking forward to have such opportunity again and again in future. Thank you very much, sir. Thank you.
I'm also very grateful to all of you for having joined and for having asked me to give this talk. Thank you very much.